Welcome to Chapter 1, Basic Ideas in the Introduction to Statistics. We will now go to Slide 2. In Chapter 1, we are going to be discussing the following pieces of information. Our learning objectives is for you to know the difference between the population and the sample, identify qualitative and quantitative variables, and to figure out which type of methods for collecting a sample is used. So, a researcher generally defines the population of interest. The population contains all of the potential information. A researcher needs to make a decision about a particular population's variables. This is a very important piece of the puzzle, right? So, obtaining information from the population is typically costly, and if we have a population with all of its information, it's only a snapshot in time, and the problem is that populations with its information tends to change with time and the variables with them. So to address the time issue of changing variables and the costs associated with collecting the information, the researcher continues to sample from the population through random methods. The samples are much smaller than the actual population, so the cost in collecting the information has been minimized. We'll talk about the size of samples needed for a study in Chapter 8 and 9. So the researcher starts out with a population of interests, and the population contains the variables that the researcher is interested in. There are two types of variables typically that the researcher is interested in. He's either interested in measurement variables like height and weight, time, pressure, those sorts of variables that have an actual measurement associated with it. And then the researcher might also be interested in variables that have labels associated with them. Labels like gender, eye color, work status, those are all labels. They don't have a number associated with it. So in collecting the sample, the researcher has two methods available in collecting information from the population. One is through a survey, which is a questionnaire, typically a questionnaire that is given to those individuals that have been selected for the sample. The other is in scientific endeavors where there is a treatment effect and you want to see the outcomes of the treatment, we typically have what's called an experiment. So those are the two methods of collecting information from the population. Now, there are what we call data collection methods, which we are going to talk about in the next slide, on how we choose individuals from the population to the sample. Once we have the surveys completed, that information goes into the sample. And again, we have two possible variables that we're collecting from the surveys or from the experiments. One are measurement variables, and the other are labels. So, from this slide, there are two major ideas that we want you to have understood. One is the population, and the population is the total number of individuals or units defined with potential information. This is what the researcher defines. And then samples are a randomly selected subset of individuals from the population that provide information through a survey or an experiment. So we actually use the samples in our studies. The population is just the beginning point in collecting the information, but it's the samples where we're going to be focusing our efforts to make decisions about the population. So now the idea here is before we can actually conduct a survey or an experiment, we have to obtain the individuals or units from the population. So the researcher has two possibilities. He can collect information from a defined population where he knows all of the individuals in the population and he can identify them. In those cases, he then assigns typically a number for each individual in the population. From that, he can then collect what we consider a simple random sampling. So, in a simple random sampling methodology of collecting samples, each individual or unit in the population is assigned a number. A random number process is used to pick a sample size n to obtain information about the population. So, typically, one example is suppose I number individuals, say I have a population of 100, and I number those individuals from 1 to 100. Well, I have, say, I have a card with numbers from 1 to 100. I put it into a box where I mix up the numbers from 1 to 100, and then I randomly choose from that box, say 30 of those numbers, right? So that's a simple random sampling process. Now, of course, the question then becomes, is it sampling with replacement or without? And that's for Chapter 5. In a stratified random sampling, the population is separated into subpopulations based on a variable, like male or female, and then each subpopulation is sampled using the simple random sampling process we discussed above. So in this case, for example, if the researcher was studying the heights of populations, he would want to separate that population by gender, male and female, so that he could accurately describe the average height of the male population versus the female population. So in that case, he would definitely use what we call a stratified random sampling. Well, what happens when the researcher does not know the population very well? There has to be a sampling method for those situations, and there is. So, if the population is not well defined, that means that we don't know all of the individuals in the population, and that could happen if you're a field biologist. You don't know all of the animals that are within a certain area, for example. You don't know how many of those individual animals exist in a certain area. So in that case, a field biologist would use something called cluster sampling. In general, cluster sampling is used for populations better defined in geographical areas. The researcher defines a geographical area or blocks within a region. Each area or block is numbered, and then a random selection of areas provides a sample. Each area is completely sampled. So, for example, if I was looking at the lizard population of New Mexico, I may want to block out, say, square miles of area throughout New Mexico, number those areas from 1 to 10,000 or whatever the square area is in New Mexico, randomly select, say, 100 of those areas, go out as a field biologist and survey that whole 1 mile square area for the lizards that I'm interested in terms of maybe finding size of the animal, finding how many there are, finding out their weight size, and so forth. Finally, another populations that's not usually well defined are typically in the retail or manufacturing process. In these cases, sometimes in the manufacturing process, we don't know the whole population because it's being manufactured. So in those cases, we use something called systematic sampling. In systematic sampling, populations are in a queuing process. That means a line in manufacturing, a line of customers entering a store, right? And in this case, the researcher randomly selects the kth individual to survey to obtain samples. So, for example, if we're looking at light bulbs and we want to measure whether the light bulb's lifetime is on average meets the requirements, we may sample every 20 light bulbs that come off the assembly line, or one every 100 that comes off the assembly line in a particular day. And we sample those and we measure how long those light bulbs last. So that's what we call systematic sampling. So once we have the individuals from the population that we're going to survey, we're interested in the types of variables or the questions that we're going to create on the survey to ask these individuals. Well, there's two types of variables that the researcher would be interested in. One is a quantitative variable, which we consider as measurement, right? So my quantitative variable is measurement, right? Measures, right? So measures could be age, income, height, weight, BMI, and there's two types of quantitative variables that we're interested in. One is called discrete, right? Discrete case are integers. Integers typically are like for age. We usually say our age in an integer number. For example, we may say our age is 22 or our age is 34. We never say we're 34 and a half. So those are discrete. So integers are discrete. And continuous quantitative variables are those that have decimals to them. So in some cases, if we have a decimal reading, we have something called a continuous variable or a continuous quantitative variable. And we'll be using those throughout this semester in the data sets that we're looking at. The other type of variable is called qualitative, and I see I've made a mistake in the actual spelling here. So qualitative. So qualitative variables or categorical variables that have a label associated with the information collected. Example is eye color, gender, work status. And qualitative variables come also in two forms. One is ordinal and one is nominal. Ordinal has a hierarchy to it. For example, if grades are assigned A, B, C, D, and F, there is a hierarchy. A is the best grade, B is the next, C is average, D is a little bit below average, and F is failure. So that's a hierarchical, ordinal, qualitative variable. So anything that can be placed in an order is called ordinal. For qualitative variables that are nominal, they have no order associated, but they are labels. Again, eye color is nominal, gender is nominal, work status is, well, I guess work status full-time may be higher than part-time, so that may be ordinal in nature. So those are the ideas about identifying the variables that we're going to be using this semester in our samples. So that concludes Chapter 1. It's kind of an overview of what we're doing this semester. In some ways, we're going to be collecting information from the population or using samples in analyzing our populations of interest. So this is one of the aspects that we're going to be looking for in this class, and we'll be discussing more in Chapter 2.